Thank you so much for inviting me. This is awesome, and this is a good opportunity to gather together, I guess, here and talk more technical a bit. I think I'm going to be more high level, but I'm more than happy to take questions lower level. I will introduce scverse for those who don't know, and talk briefly about what we're doing and how we're trying to prepare this era of AI. Because I figured that a lot of people use tools that they belong in our organization, they're maintained by us, but they're not necessarily aware who we are and what we're doing. It was a very good introduction before, because we mentioned the AI boom that just passed, and now that the data boom that was announced by the latest JPM. And my thinking is, okay, people are generating massive amounts of data. What is coming next? It should be the infrastructure, the data loading boom. How do you load this data? We have all these GPUs, CPUs nowadays, all this infrastructure happening, and a huge amount of data, but these necessarily are not talking to each other, not properly. So yeah, scverse is in this space. So I'm going to go a bit back and also briefly talk about 15 years of history, more or less. I'm Michaela. I'm a postdoc at Genentech in the morning. That's my actual job at Marioni, John Marioni's lab. And I'm a steering council and core member at scverse, which is the evening job the last 4 years, after my morning one. Doesn't pay my rent, otherwise, in other words. Story time, why do we exist? So 13 years ago, for those who don't know, there was the Methods of the Year was awarded by Nature Methods, and it was the single-cell RNA sequencing, which was not when it was actually happened. It was when it was established enough to be awarded as the Methods of the Year. And very soon after that, like 7 years later, 2020, we have the Spatial Transcriptomics as the Method of the Year by Nature Methods. And basically what was in the space, I'm trying to kind of move briefly from that because it's history, that's very interesting. So in the space was bioconductor, right? There was already most of the omics data history was in R. Statistics were there, and actually our story comes from there. We copy a lot of information from them. It was pioneers. So they have all these summarized experiment, multi-assay experiments, single-cell experiments. Because the space was kind of fragmented, this starts showing the way of how do we gather the community, how do we gather the tools, and how do we take advantage of this data. So in a similar effort, there was UnData happened 2018, and actually ScamPy even before that, which was 2017, published in 2018, but existed. It was an effort from Alex Wolf and Thijs lab in Munich because they didn't like R back then. No hard feelings. I like both. But it was an effort. And they were trying to establish there the ScamPy, which was a similar effort in R. But it was a very fragmented space. And again, you basically create another issue where you added an extra tool that communicates with nothing. You had to translate the data and transfer them and everything. So UnData, which is annotated data, came to save this and create an object where you load your data properly and you store all the information you need. And soon after that, 2020 officially we were launched, and it was the scverse, which is basically the ecosystem around that. And I will explain, this is a very preliminary, the first publication we had where we're establishing the ecosystem, and it's already outdated because already the data structures are more than this one. But it kind of captures what we do and who we are. In our heart is the data structures. So we have the UnData for single cells and MU data for multiomics, and nowadays spatial data too for spatial data. And the outside layer is the scverse core packages. You may recognize some of them, but basically the idea is that they are all the preliminary steps to analyze the data that they are coming from the data structures. So the first step is data loaders, data structures. Second step, core packages to analyze this data, the preliminary analysis. So the difference with the ecosystem is that these two layers we maintain and we develop ourselves. So the second part, the ecosystem, is all the tools that they actually comply with our code of conduct. They have some very specific minimal requirements to take advantage of our data structures, basically, and be in the space of single cells. But they are maintained by the developers. We only work with them just to make sure that it's easily maintained, the tools. And if some tools become very important for the community, we can actually take over and put them in the core, in collaboration always with them. And the outside part is always the community. We're very community-driven. This is how we exist, and we do all the events and everything. So data structures, I introduced them. This is, you're familiar with the space. Undata annotated is basically the main way to load your data in an object right now in Python. Undata. Mudata is a dictionary of undatas, so as many modalities as you like. They just share metadata, and they're basically structured and connected together. And spatial data is kind of interesting because it has undata, the tables, the expression values are in an undata object, but you can also load all the pointers, like the images from raw images are and everything. You can all load it in spatial data, and they are layered and stacked, so you can analyze them together, and you can maintain all the information. And the core packages that we have is right now, because we expanded recently also, ScanPy for all the single cell preprocessing up to visualization, basically, even differential expressed genes. First steps you can do with ScanPy. SquidPy for spatial data, similar idea. The same expansion of preprocessing of multiomics data is the muon. SCVI tools. Valentine is one of the main contributors, not directly probably in SCVI, right? But all the ecosystem around that. So it's the variance inference tools for all things single cells. Snap attack for ATAC-seq, decoupler for inference, gene inference, ShearPy for immune repertoire, PertPy for perturbation data, and Rapids, which is very cool. Our Rapid single cell is the wrapper of ScanPy, basically, but takes advantage of GPUs. We're talking about from hours to seconds. It's insane. It's one of our biggest tools at the moment, and we love it, and it's a collaboration with NVIDIA. But as I mentioned, a big part of scverse, for those who are interested in getting involved or getting to know us, is the community, because we're a community-led organization. And sometimes there's this misconception, because most of us, we're developers there, but basically developing and writing code is only one thing, and it doesn't come only from the developers, especially these days with agents. Writing the code is probably the least of our worries. But we do all these hackathons where a community gathers together and solves all issues, PRs, pull requests in a way that we have open. And then we have all of the support on GitHub, tutorials, you name it, we do it. Atlases we maintain. And basically, we're inviting always the scientific community to contribute in all the ways. And you can do it from any position you have, either you're in company or academia, it doesn't matter. Any contribution helps in an open source organization. Some stats, just to flex a bit, because that's necessary. So, I mean, here we saw that we have more than, I don't believe that GitHub stars is a great way to evaluate tools. That's fine, though. We don't have better metrics. So Undata, basically, you can see our biggest one is ScanPy, of course, makes sense. The GitHub stars, and also spatial data that are coming up, because it's a more recent addition. SCVI tools, of course, huge impact in the field. But the most important is, and this is kind of outdated now, we have more than a million monthly downloads of all of our tools, which is insane. And I will explain later why it's insane and what is the main issue we're facing. So this is increasing a lot. And nowadays with agents, please don't submit PRs that you haven't reviewed first. It's an issue, okay? Zulip, for those who don't know and would like to find us, Zulip is, I guess you know Slack. Zulip is Slack for cool kids, and we're the cool kids, so we do Zulip. Zulip is awesome. There are a lot of reasons we're there. A lot of open source communities are moving there right now. Bioconductor is there, NumFocus is there, Napari is there. So you have to join us. We have like way more right now than 1,000 registered users and 1 million messages read, which basically means that we have like around 100, 150 people always interacting. So if you have any kind of question, there's going to be someone that will pick it up and will respond. And the developers are always there. So if you have a question about a tool, probably you will find them live to answer your questions. It's not a forum necessarily, but you can also use it like that. Right, flexing a bit more, just the things we've done with scverse, because the last 2 years we've been working very hard, I think, and we're producing very hard. We have collaborations with a lot of academic labs, and we are publishing papers in collaboration with them. Biocontext AI, that we'll briefly mention a bit later, is one of them. Undata R, so now the R users have no excuse anymore. They can use Undata if they want, and they can transfer all their data back and forth in languages. We are in every space. We're doing it in Rust soon. Spatial proteomics, PertPy, and other spatial proteomics is 2 different papers. Ignore. Yeah, and a second one. I mean, we have this consortium is very live. So we have a lot of efforts, and I like, before I go to the final topic, which is kind of more the AI and how do we make ready and the new efforts, I would like always to introduce the team somewhere in the middle where I kind of have attention or I get back the attention. The highlighted ones were the steering council. That doesn't mean anything, of course, for you. It's like we're part of the core. You can find a lot of us nowadays in, around here, San Francisco, and the rest, they are all over the world. This is an awesome team who has been together for years. And of course, our management committee, which helps us and funds most of the things so far. You may recognize some names, but basically we write grants together, they advise us during the process, and we meet very often with them. And finally, we have the advisory committee, which we meet a couple of times per year, and best case scenario. And if we meet, they are very, very helpful because they steer more of the directions that we have. But the most important is the contributors and the community, because I mean it for an open source, the most important is the community, and any kind of contribution really matters. Just for me to mention here already that from all the people you see, only 2 are full-time employees at scverse. The rest were volunteers. We don't get paid for that. It's very important for the rest. So where we are and what are we doing later? So I kind of skipped the first part. They are always very important. We have been doing them 2025, 2026, probably since we started 2022. We've been trying. We scaled more than 100 million cells to analyze. We do multimodal, any modality. Nowadays, we basically try to do all things single cell. We started as transcriptomics, but in reality, single cell is resolution. We need all the modalities to understand the cells, to understand the drugs, how they interact. It's not only one version, especially the virtual cell that is a big thing nowadays. If you only consider RNA, it's a virtual mRNA now. It's not a virtual cell, in my opinion. So I think multi-omics is a big thing. We always care about benchmarking, and especially now we have a bigger effort coming up on benchmarking, and of course user accessibility. But lately we want to be the cool kids, as I said. So we always care about agents and scverse-specific MCPs. We've been collaborating with Anthropic, that they have been sponsoring and giving us some of the credits that they use for they give for open source, and some power, mental power, I would say, not engineer power yet, but we've been discussing. And we have Into Context AI that I mentioned before, which is a company right now that they basically create all type of MCPs. It's kind of an effort between academia and non-profit, but they are turning into profit. So don't quote me that. Can cut it later, right? Right, in case it's too soon. And here is what I want to mention, what we're doing, because the idea is that we're not creating really, we are creating MCPs on data MCPs, or we collaborate on these things, but this is not what we're doing. This is not, we're infrastructure, right? We're trying to enable and make AI-ready data structures and data for people to be able to use them with LLMs. So the first thing that is kind of obvious, you would think, is that we need to make the data ready for AI, right? So the first thing we did is that we're working on this Unbatch, which is streaming data loaders in order to be able to saturate the models while training, from reading them from disk. Because as we said before, the models become bigger and bigger, so you cannot load them in memory, and you need to be able to train efficiently by loading from the disk. And that's kind of a solved problem. There was an ARC Institute effort recently in a similar vibe. So we did something very similar. Ours is undata tailored, so it should work directly with the whole ecosystem. But this is not the only thing that's important. This is happening, and it has to be a collaboration with companies as well, in my opinion, that they produce big data sets. The next thing that we care a lot is the metadata. So the data are important, and you can train models, and that's awesome, but it's always going to reflect the data, the annotation, right? How many things you know about the data and how well they are annotated. So we are building croissant, awesome, metadata descriptors, which is a machine learning format, anyway, documentation for this job. So basically, now the tools can actually take advantage directly. They can store the metadata properly, and they can be read by the LLMs and agents, and not only, also in general machine learning models. And finally, we want to connect also everything with the existing cell ontology, the existing ontologies, especially MBL-ABI is our main target. We have cell ontology for cell types, Uberon for analysis, for anatomy, and disease ontology for conditions. In general, the main issue we solved with Unbatch is the input-output, because you end up a lot of times basically spending more time on loading your data than actually training them on the models. So that was the first goal. It's kind of solved. We're still working on it, but Unbatch is live. It exists. You can use it. And then another big effort, as I mentioned before, of course, is the actual MCPs and the agentic workflows. We already have an on-data MCP. You can use Claude. You can read it. It's live, or any MCP tailored anyway method. So you can use already and query your data. You can understand basically from on-data directly. And we are expanding this to spatial data, ScanPy, SquidPy, PertPy, basically all of our core packages. And this is indeed the collaboration that I was referring before with Anthropic, with the credits, and Biocontext AI, that they have released a lot of MCPs already. So now you can basically use directly on data, as I said, and continue with the rest when they come. But another thing that we care, I mean, not just us, and as I said, this is not our main goal, is how can you create a whole workflow, right? How can you give an on-data object now and basically get out of it, in our case, an scverse workflow, you would say, like use the scverse tools, but it doesn't matter. How can you get out the whole, do the batch correction, annotation, clustering, everything, and get the output to UMAP? I don't know. This is the very minimal workflow I can think of, or cell-cell communication. You can. So we're thinking of that. How can we tailor all the scverse tools and order them, or maybe pre-decide? It's kind of a tricky problem to solve, especially as a community, and it's not exactly what we're doing. But this is also in collaboration with Biocontext AI. Finally, another very, very exciting for the single-cell community thing that we've been doing is that we are rewriting, it's led by Rob Patry and James Ferguson. We're writing Star Aligner, which is, I don't know how many years. I don't know. I don't remember. It was written in C++ 98, the code base, and it's like more than 50K citations. Is the main tool for aligning a single-cell dataset, and it's purely unmaintained at the moment. So what we're doing is that it's called Star Aligner. It's also live. You can find it. And basically, we translate with Claude from Python, from C++ to Rust. And we already established that it kind of works. We're still running things. We need to make sure. But it's exciting. It's a collaboration with NVIDIA for the GPU accelerated components. But the cool thing is that it's a real-world example how we can use LLMs. I don't want to advertise only Claude, of course, but we are collaborating with them. And the main thing is, one of the problems that we face is the maintenance of tools, right? That's what we're trying to do, and that's what we're trying to provide for the community, but it takes a lot of effort. And this actually is the first demonstration that we may be able to do it fast, like quickly iterate, quickly maintain, and successfully, with minimal review. That's what we're trying to test here. So far it works. We'll let you know if things change. But this is exciting, actually, for us and for open source communities, I think, in general. So it's kind of a summary now, what's working. I briefly hyped our things. Unbatch works. On-data MCP is live. You can use it. Star Aligner also is happening. If there are any issues, you can also go on GitHub and contribute if you want. And Biocontext AI basically has created this skill to MCP breeds, where basically whatever MCPs exist, you can already take them as anything that reads skills. You can load them, the MCPs to the skills, and the skills can be loaded on the LLMs. It's also in collaboration with Claude, with Anthropic. But everything is not solved. And here I decided to put some open challenges. That doesn't mean I have the answer or I'm working towards that, just ideas. So, OK, of course, the first question is, how do we know biology is correct? I don't think it was solved as a humans. We solved that, and now the problem is the agents. I think this is a pure question. You run the whole workflow, and how do you know what you're running, the parameters you've chosen, what you see in the UMAP, or any other plot you generate is correct? The only issue now is kind of that, I don't know about you, but I don't really write code anymore in this analysis. There's no way I'm actually going to type anything in ScanPy anymore. They do it for me. And the problem is that you cannot review so much anymore. Like, the agents can decide to cluster, annotate, and then come up with some solutions, right? How do you even know? What is the benchmark? Not benchmark in a sense of biology necessarily, but how do I assess this in a way that the user will actually go and read the results? That's the main question. And then we have the issue of a lot of things now they're demonstrating some technologies, especially with agents and the pipelines. And then you have similar technologies. Here I demonstrate, I don't know, Visium, Murphy, Sinium, Cosmix, and Codex, that they don't, all the pipelines work from each other to each other, even though they should. They produce similar results. And that's in my opinion, an issue because you want as general as possible, and then you need to think, why does it work? If the data are similar, why does it work in one and not in the other one, in the other modality? Metadata standardization at scale, as I said, garbage in, garbage out is not, I'm not the first one saying it, so it's very important, I think, to be able to curate the metadata. And as I mentioned, a big issue with all of these things is that a lot of things are relying on open source structures. Think about, we're talking about 1 million downloads per month, and a lot of companies, they're building on data and ScamPy. I bet among all of you, probably some of you use it, if not all of you. And as I said, there are only 2 employees, and the rest were volunteers. So this is not very sustainable, and that's a big issue and an open challenge. We're writing grants and everything, but yeah. And that's the final slide that I kind of repeat everything more or less, what we need next. I think I said it, I'm not gonna bother you more. I think is the data, they should be ready immediately to be read by the machines with metadata. They should be able to be loaded properly with GPUs, especially take advantage of GPUs and not just use them and like waste time on, the bottleneck is input-output, and then you have GPUs just waiting, then it's just a waste. So all of these things should be produced directly and documented for machines. And then basically we need verification, like we need reproducible agents. Right now all of us use them, but very individually, and you can't even share them and say, okay, to your collaborator, here it is, reproduce it entirely, or you're not sure, basically. And finally, the sustainable. I would love the companies that they use us to find ways, we can find together the ways to sustain and sponsor us and help us grow and sustain ourselves. And I have a last message, because this event is single cells, and our history is single cell RNA-seq. But this is food for thought. We've been expanding, as I said before, we're doing all things single cells, not just single cell resolution. Modalities are important. And lately, my obsession, you probably noticed from the papers that we published the last year, is proteins, because 70% of the drugs, they're actually predicted and validated on proteins, not on RNA. RNA is great, but just maybe not enough. And think beyond. That's food for thought. Just on. Call for action. Just contribute to the community. I mentioned everything. You don't need more than that. Anything you do is, even joining a hackathon is very important. And we have meetings also every other Tuesday. You should have introduced that, Valentine. And that's it. And thank you so much for inviting me. And I'm open for questions. How they look like? So the croissant metadata. So it's a very standardized, like a YAML almost file. It's standardized, but it's standardized in a sense that it's easy to be read by ML, machine learning models directly. And it's already pre-decided that it's tailored to the, if I can use English words for that. So it has predefined sections, and it's not very hard to use it. The hard thing with the metadata is to decide what you ask from the community to always have. We don't have that so standardized in single cell RNA-seq. Like, you should be able to ask, like, I don't know, Chromium, which version of which kit of 10x you used and everything to be always stored. And that's one of the efforts we're doing, to pre-decide what everyone needs to always claim in an undata object in order to be pre-processed. What do you need to store? And that has been a big issue, because now that we're moving to different modalities, each modality has their own requirements. So we're trying to find the bare minimum for everything.
How standard do you think you're going to be able to make metadata? Because I feel like it's so hard with all the different types of experiments people are doing to be able to do that.
Yeah. So the question is, how standard can we make the metadata, right? Yeah, we can't. So the idea of scverse in general is to always provide kind of best practices, right? We're infrastructure, and there's not one way to do things. I think that the impact we have right now in the community can be helpful to establish a way of doing it. And then if everyone does the same, I think that's at least how I see it, more or less. But if you have any suggestions, you are more than welcome to give us.
The idea of like rewriting libraries like Star with Rust sounds very promising. Like you said, it's really hard to maintain a lot of these tools. It's super interesting. What performance improvement did you see after rewriting it in Rust versus how it originally performed?
Yeah, that's a good question. I don't have the numbers entirely. The main thing it solves for us is not so much the speed, for example, or how accurate it is that we can maintain it now, right? Because the C++ 98 code base, it was not maintainable, and it was not maintained either. So probably you get some performance boosts, for sure. I don't have the numbers, but I can look it up. But for us, it solves the maintenance problem, basically, as a community in general. Yeah.
Yeah.
Sorry, one last question.
Yeah.
How do you bridge the gap between metadata that's grounded in ontology versus, say, author-provided annotations or metadata? Because sometimes a newer paper might discover like a new cell state within a cell type, for example, but that might not necessarily exist in an ontology space. So what do you do in such edge cases?
Right, that's a very good question. One of the biggest discussions we have in the group is basically his question, which was, how do you solve? So annotations, you have already ontologies with some specific annotations, but most of the papers, basically, they discover new subtypes that they don't exist in the annotation, right? There's no entry. And the idea is, because we don't solve that, right? We offer solutions on how you can store this information properly. The idea is to connect the ontologies. So if it's actually justified, because I have also the feeling, I don't want to judge the science at all, but I have the feeling that a lot of times people claim things that they are, a set of markers that if you were looking in this ontology, maybe it was similar or whatever. So if there are some thresholds on some markers, it can be a new entry on your own data, and then it should be linked and published also in the ontology. So you populate an ontology, which is curated, because the EMBL EBI cell ontology is curated, right? So something like that we are thinking. But you should, as user, you should always have the flexibility to first use what exists, and then add anything you need on your data. And if you don't want to publish it and connect it with the ontology, that's up to you. But yeah, for us it's a dictionary, so you can have new entries. That's not an issue. Yeah. Cool. Thank you.
Thank you very much. We will probably donate. We seem to be active users of all the tools.
Active users?
Yeah.
He said we'll probably donate.
Yeah, I didn't realize that you guys were taking contributions.
Yeah, for sure. Please contribute. Just not completely LLM generated PRs.
I meant more funding.
Funding. Yeah. Oh, you didn't know? Oh my God. So.
I didn't know there were only 2 full-time employees.
So yes, we are getting sponsorships, and we're changing now, for those who want to know, our non-profit. So we're going to have a board with companies, so we can talk.
Yeah, definitely.